Konnichiwa. Good afternoon. It is an honor for me to deliver this public lecture on behalf of the Worldwide Protein Data Bank Foundation. Today I will explain some of the ways in which the Protein Data Bank, the PDB, is contributing to the search for new medicines.
皆さんこんにちは。wwPDB トラッカーを代表してこの一般講演会で講演することは大変光栄です。本日はプロテインデータバンクの新しい学研究に対する貢献について説明をいたしたいと思います。
In 1953, Doctors Watson and Crick, working at Cambridge University in the country of my birth, England, published their landmark paper in the journal Nature describing a molecular model of the DNA double helix. The Watson-Crick double helix model, shown between the two scientists, made a critical difference to our field because it explained in molecular terms how DNA can serve as the repository for genetic information passed from one generation to the next. The Watson-Crick model is just that. It is a model. A low-resolution view of the structure of DNA. The PDB contains many atomic resolution structures of DNA, the first of which, produced by X-ray crystallography, is shown here on the right panel. DNA works in harmony with ribonucleic acid, or RNA, and proteins to make life possible as we know it. If you remember only one thing from my lecture, please remember, DNA makes RNA, makes protein. DNA is our genetic blueprint. Messenger RNA, mRNA, is the intermediary between the blueprint and the proteins which actually make life possible. The process of reading the DNA blueprint and making the messenger RNA is called transcription. A molecular machine, RNA polymerase, synthesizes the messenger RNA from the DNA. Messenger RNA is also made up of 4 bases, G, C, A, and U, which takes the place of T. In transcription, the 2 DNA strands are separated by RNA polymerase. The messenger RNA is synthesized by linking the 4 RNA bases together in the correct order directed by the DNA template. A on RNA pairs with T on DNA, U on RNA pairs with A on DNA, C on RNA pairs with G on DNA, and G on RNA pairs with C on DNA, base pairing. Translation of the messenger RNA to the protein determines your phenotype, what we are, who we are. Messenger RNA serves as the working drawing between the DNA genetic blueprint and the protein. Each messenger RNA makes a different protein. The process of making protein from RNA is called translation. Another molecular machine called the ribosome decodes the messenger RNA and synthesizes the protein chain, which is composed of 20 different types of amino acid. This schematic shows the ribosome translating the collagen mRNA to make the collagen protein, a repeat of glycine, proline, proline, glycine, proline, proline. You will see in the next slide that this triplet repeat gives collagen a very interesting 3-dimensional structure that explains how it works in our bodies. Proteins are the building blocks of cells and molecular machines that support life. Some proteins are found in every cell, like the proteins required to make RNA from DNA, and the proteins required to make proteins from mRNA. Other proteins are more specialized and occur in only one type of tissue. Before I show you 2 proteins that carry out similar functions in very different tissues, I promise to show you collagen at work. The amino acid sequence of collagen glycine, proline, proline, causes it to assemble in a rope-like triple helical structure, shown here on the right. Thousands and thousands of these triple helices line up to create molecular rope, the tendons in our bodies, like the tendon in the palm of your hand. You can feel it. Collagen is called a fibrous protein because it assembles into fibrils. Most proteins are globular, meaning the protein chain folds up into a ball. Myoglobin on the left is the most abundant protein in muscle cells, where it stores oxygen, making it available for us to flex our muscles. The myoglobin protein chain folds around a small flat molecule with an iron atom in the middle called a heme group. Oxygen binds to the heme iron until it is needed by the muscle cell. Hemoglobin, shown on the right, is the most abundant protein in red blood cells, where it carries oxygen from the lung to the rest of the body. Hemoglobin consists of 4 chains, 2 alpha and 2 beta. Each of these 2 types of hemoglobin chains has the same fold as myoglobin, with the heme group in the middle. Comparing the structures of myoglobin and hemoglobin, both of which can be downloaded from the protein data bank, you can see that these 2 proteins with similar biological function, reversible binding of oxygen, have similar 3-dimensional structures. In biology, function follows form. In 1928, Alexander Fleming, working in England, accidentally discovered the antibiotic penicillin. The story is simple. Fleming was studying Staphylococcus aureus, a bacterium that causes infections of the skin. He was growing these bacteria on a petri dish. One dish became contaminated with a fungus called Penicillium notatum. Instead of throwing the petri dish away, he looked at it very carefully and realized that bacteria were not growing in the vicinity of the penicillin mold. As the famous French scientist Louis Pasteur said, nature favors the prepared mind. Fleming's mind was prepared. He realized that the fungus was secreting a substance that killed bacteria. Penicillin works by binding irreversibly to the DD transpeptidase target. The PDB contains many structures showing penicillin in action. Here, we're looking at penicillin binding to the DD transpeptidase of Streptomyces. The active site of the DD transpeptidase is shown here with penicillin bound. When penicillin binds to the active site, it forms a covalent bond with the enzyme, killing its chemical function. With the DD transpeptidase inactivated, the bacterium cannot make the Kuzari armor, and without the armor, the bacteria are fragile and die quickly. Even before penicillin became widely available, Abraham and Chaim, working at Oxford University, my alma mater, discovered an enzyme, a bacterial enzyme, that inactivates penicillin. They discovered beta-lactamases, which cut the 4-membered beta-lactam ring, shown here. Once this bond is cut, the antibiotic is no longer active. Unfortunately, nature is winning the bacterial antibiotic arms race. The New Delhi superbugs you have read about in the newspaper carry a gene encoding a new type of beta-lactamase. This enzyme does not resemble the DD transpeptidase. Clavulanic acid does not work on NMD1, shown here on the right with an inactivated ampicillin bound in the active site. Scientists now have a new drug target, NDM1. one of the students in the audience could discover a drug that works against NDM1. Please study hard, stay in science. Remember, nature favors the prepared mind. As promised, I'll now present an example of targeted drug discovery for treatment of a white blood cell cancer. The cancer is chronic myeloid leukemia, or CML. These large white blood cells proliferate in bone marrow and blood, causing the disease. The drug is Gleevec. Before Gleevec, CML was fatal, 100% fatal. 5-year survival on Gleevec is greater than 90%. One pill of Gleevec makes all the difference. Not all patients were helped by Gleevec. Some have cancers that are resistant to Gleevec. DNA sequencing of these resistant cancers showed that they have mutations in their BCR-ABL genes that block binding of Gleevec. The locations of these mutations are indicated in orange on the structure of the BCR-ABL fusion protein. The scientists and doctors studying chronic myeloid leukemia said, we need a new drug. Using their knowledge of the 3D structure of the BCR-ABL fusion protein and the locations of the mutations causing resistance, scientists at Novartis modified the chemical structure of Gleevec to make a new drug, nilotinib. Nilotinib works against most of the resistance mutations. It's also called Tasigna, and it's now the drug of choice for treating chronic myeloid leukemia. The scientific heroes of this epic story were honored by your emperor earlier this year. Doctors Rowley, Leiden, and Drucker received the 2012 Japan Prize for the discovery of Gleevec. I have some final words of advice. Please remember, DNA makes RNA makes protein, and in biology, function follows form. Some career advice for the students. Today's biomedical research combines biology and medicine with chemistry, physics, mathematics, and computer science. Many ways to contribute. どうもありがとうございます。